Both these chapters are talking about membranes, membrane structure, and membrane function in the case of Chapter 12. And some of this should be review, hopefully, from general bio. But I just wanted to go over membranes, the lipids that make up the structure of the membranes, the properties that biological membranes have, as described by the fluid mosaic model, and the proteins that are a large component of membrane function. Okay, so membranes in eukaryotic cells, there are a lot of membranes. What about prokaryotic cells? Bacteria. Just one, which is the plasma membrane. Okay, so eukaryotic cells have phospholipid bilayers. Okay, so a membrane is a phospholipid bilayer, 2 layers of lipids. And that applies to all the membranes of all the membrane-bound organelles, as well as the cell membrane. And these are important barriers, right? Basically, they allow the cell to compartmentalize the functions it needs to achieve. And in the case of more simple organisms like bacteria, it allows the cell to separate itself from that chaos of outside the cell, that disorder that we talked about with the first and second law of thermodynamics. What does it mean to compartmentalize? It means that certain molecules can be located in certain sites within the cell. And the concentration of those molecules can be controlled. Okay, so a cell wants to import a lot of food, a lot of glucose, for example, needs to get into a bacterium in order for it to make energy. So even if there's not very much glucose on the outside of the cell wants to still transport that glucose inside the cell. Now, some things can move freely in and out of the cell, while other things need some help, and that's where the proteins in the lipid bilayer come into play. So let's first look at the lipids. Lipids have the general structure of being amphipathic, which means they have part of the molecule that's hydrophobic and part that's hydrophobic and part that's hydrophilic. Okay, so the head is hydrophilic. What does that mean? Is it water-hating or water-loving? Water-loving, okay? The tails are hydrophobic. They're basically carbon-hydrogen chains. Some of them have no double bonds, whereas others have a double bond between the carbons, causing a kink, which makes a little bit of space between the lipids. Now remember, lipids, a single lipid is a subunit of a membrane, but the lipids don't covalently link like other macromolecules. They aggregate together. And actually their amphipathic structure helps this to happen, to be energetically favorable and happen naturally within an aqueous environment, which we know both inside and outside of a cell are full of water and aqueous environments. So let's just look a little closer at the details of the structure of the lipid, and then we'll go on to why they tend to orient the way they do. So the head is made up always of glycerol and a phosphate group, and then it could have another group. The most common phospholipid in our membranes is phosphatidylcholine, which has a choline attached to the phosphate. And then from the glycerol, so a triglyceride is just glycerol with 3 fatty acid tails, but in the case of a phospholipid, because one of the sites on the glycerol is bound to phosphate, there's only 2 free sites available for binding the fatty acid tails. So all phospholipids have 2 tails and a polar head. And as I mentioned, some of those tails have a double bond, and that double bond can actually change depending on the conditions that the cell is in. Okay, so the most common phospholipid is phosphatidylcholine. And when these lipids aggregate together, because of this 2 ends, the head being water-loving and the tail being water-hating, they just naturally form a bilayer with the water-hating parts towards each other. So the fatty acid tails orient towards each other, whereas the heads orient towards the aqueous solution, either outside of the cell or inside of the cell. Now, even further than that, if we had a plane of phospholipids, there would still be exposure of those tails along the edge to water. Okay? But that's an unfavorable condition because being nonpolar, there's no way for these tails to interact with water. So they really naturally move away from it. So what naturally happens is that a sealed compartment forms. Okay? So in the case of a small amount of phospholipid, a small bilayer, it could be as small as a vesicle inside the cell. But the plasma membrane itself always maintains this orientation and the shape that allows for the, rather than a planar shape, a sealed compartment to be maintained. Yes, Lynette? So they always flip so that the heads are on the water side and the tails are buried in the so here the red is the heads, the white is the layer where you see the tails. So they'll always flip into a bilayer. If you just think of oils themselves, just oils like triglycerides or vegetable oil, if you put your salad oil in water, it immediately coalesces, right? And that's because it's hydrophobic. It doesn't have a polar head. It just stays away. It forms these little droplets of oil within the water. When your salad dressing separates, it's because the oil is hydrophobic. Well, before the heads are formed, they're not in a bilayer. So the lipids are actually made in the ER, and then they become part of the ER membrane, and then they're distributed to the other compartments within the cell. So if you just artificially do this inside a tube of water, you'll get a liposome. But inside the cell, this also happens when compartments, for example, the Golgi has a piece of it pull off, it will always form a vesicle or a circular compartment within the cell. It will never become planar. So in that way, the cell is able to maintain the integrity of each compartment, right? It's worked so hard to get certain solutes in certain places within the cell, and it can be maintained simply by the properties of these lipids. So even though the orientation will always spontaneously be the heads exposed to the aqueous side and the tails flipped in towards each other, there is movement of the lipids within a membrane. And they can move either side to side, back and forth, along one of the layers of that bilayer. or they can rotate in place or spin kind of in place. But they very, very rarely will switch from one layer to the next. It's not energetically favorable. So this movement or diffusion of phospholipids within a membrane is what we call the fluid mosaic model. Okay, so it's fluid, constantly changing. There are conditions that can hold certain parts of the membrane in place or cluster certain proteins into one area of the membrane, but there's always movement within the membranes, whether they're internal organelles or the cell membrane. Now, in addition to phosphatidylcholine, there's another phospholipid that's found less frequently, but also in eukaryotic cells, phosphatidylserine. So the only difference in the structure, you still got the two tails, the glycerol, the phosphate group, and now serine. There's also in animal cells, such as our own human cells, cholesterol, which, as you can see, the hydrocarbon chain is much shorter here and gives a more rigid structure to the membrane because of this inflexibility. So there are no double bonds here, so it can pack in tight, fill in the gaps that are created by the phosphatidylcholine, which has the bent, one of the hydrocarbon tails is bent. Okay, and also it's a different kind of lipid. It's a different kind of fat known as sterol because it has these ringed structures attached to the hydrocarbon tail. And then finally, glycolipids, which are lipids that have a sugar on them. In this case, galactose. Okay, so they still have the hydrocarbon tail structure that will aggregate with other lipids, with the phospholipids in the lipid bilayer. But instead of the serine or the choline, it has a sugar. It's still polar, and so is cholesterol. It's still polar, so it still orients itself where the tails, so they're still amphipathic, that they have this polar region and the non-polar region, which helps them to orient in a certain direction, right, with the head group out towards the aqueous, outside of the cell or the inside of the cell, and the tails to orient towards the other tails from the other side of the bilayer. And in the case of these glycolipids, there are actually a lot in eukaryotic cells. They account for the ability of the cells to move. So in certain types of cells, for example, white blood cells, that would need to move from the blood to the tissue, like a neutrophil that will extravasate beyond the endothelial layer into an area of tissue where there's an infection. These slimy or slippery sugar molecules that coat the outside of the cell membrane as part of the glycolipid content of the cell membrane help it to make those movements, as well as specific receptors that interact. So lectin receptors that will interact with lectins and help to slow the movement of the cells through the blood. So cholesterol lends not just rigidity, but strength to the plasma membrane. And as I mentioned, you can see here, it kind of fills in those gaps that occur between the phosphatidylcholine components of the membrane because of their hydrocarbon tail that's unsaturated. So as I mentioned, there's a fluidity or movement to the membrane on both sides, but rarely do you have a phospholipid from one side moving to the other side of the bilayer. And that allows for the membranes to be asymmetrical so that those glycolipids can be oriented to the outside of the cell where they're useful and necessary, but not to the inside of the cell. So there is a sidedness or asymmetry to not just the cell membrane, but also internal membranes of the organelles in the case of eukaryotic cells. So how do these glycolipids, cholesterols, phospholipids that are made in the ER get to, one, the right side of the membrane, and two, into other membranes in other compartments of the cell? So when they're synthesized in the lumen, they actually are added to one side of the membrane of the endoplasmic reticulum that faces towards the cytosol. So you actually get a thickening of just that side, but then an enzyme helps to catalyze the transfer of that flip-flop so that there's an even or symmetrical growth of both halves of that membrane. And then pinching off of that membrane and transport to the next compartment, the Golgi, and then from the Golgi to the plasma membrane will allow the cell to grow, to divide. When cell reproduction occurs, it allows it to make a new cell plasma membrane, etc. Yes? Vesicles are these bits of membrane bilayers that are pinching off. So it's the vesicles that are mediating transport not just of the lipids, but anything that's pinched off inside the lumen of that vesicle, as we'll see in Chapter 15 with protein transport in the cell. Okay, so yes, membrane-enclosed organelles can redistribute or be formed as a result of vesicle movement, or they can transfer lipids or their contents to the plasma membrane, or even if they have some molecules that they want to secrete, they can be released to the outside of the cell. Okay, waste. In the case of microorganisms, they can secrete toxins. In the case of our own cells, they can secrete mucus for the mucous membranes, etc. So phagocytosis is if you just flip the arrows the other way, and instead of being an organelle, well, it would be an organelle, but it would be not the Golgi, but it would be a lysosome or an endosome. That would be the vesicle pinching off from the membrane, coming into the cell, would fuse with, and then become part of that compartment. Yep, so both works both ways, in and out, bringing things in and out of the cell. But basically, this is how the cell grows, is by shipping the lipids made in the ER through the Golgi to the plasma membrane. So besides lipids, membranes have a large component of proteins. So actually they're about 20% proteins. By weight, it's about half by weight, but if you look at, because the lipids are so much smaller, they're about 50 lipids per one membrane protein. So there are a lot of proteins, and so naturally that would give an indication that proteins have an important role in our membranes. And the proteins are really what help the cell to control selectively what gets in and out. Because remember, the lipid bilayer is only going to let certain small uncharged molecules readily diffuse across it. So these protein molecules are also an important component of the membranes. Some of these proteins will span the entire lipid bilayer and help with functions in the cell like transport or to hold things in certain place within the membrane. For example, attaching to the cytoskeleton to give a certain shape. Or they could be receptors that are able to bind extracellular ligands and signal internally so that the cell can respond to changes in its environment. Or they can be enzymes that help to catalyze reactions within the cell. And these proteins have different structures. So we already went over the tertiary structure of proteins. In the case of the transport proteins, whether they're channels or transporters, they span the membrane many times. Okay, so we call them transmembrane proteins because they have regions within their amino acid structure that either form alpha helices or beta pleated sheets, forming a pore or a channel through which solutes can move. And they're also even just for signaling. So an example of that I gave in the previous slide of the different functions, these would all be transmembrane proteins because they all span the entire membrane. But they would have a different number of times that they span the membrane, whether they're just a signaling molecule or whether they're a channel through which solutes will pass. There can be proteins that are only associated with the one or the other side. Those are the membrane-associated proteins. They can be associated with the membrane by the protein structure itself. So remember, all of these interactions with the membrane are through non-covalent interactions with the amino acids in that area or that region of the protein. So they can be membrane associated on one or the other side. They can be associated through a lipid moiety or part of the protein that's lipid. Or they can be a protein that holds a protein in the vicinity of the membrane in the case of proteins that have quaternary structure, right? So multi-subunited proteins. So we're going to talk in more detail about transmembrane proteins that have an important function of moving those solutes that are not able to readily diffuse through the membrane in or out of the cell. And that's covered in Chapter 12. So let's look at the structure in a little more detail. So remember that it's really the amino acids that the way that they're oriented in alpha helix. Okay, the side chains are pointing out. Okay, so in this region, would you expect the side chains to be the transmembrane spanning region of that protein? Would they be polar or nonpolar? Okay, they're adjacent to the hydrophobic tails of the lipid bilayer, so they would be nonpolar, right? If they're polar, they would tend to naturally orient towards the outside of the membrane. In the case of multiple membrane-spanning transmembrane proteins, though, it can be a mix of, here in green, nonpolar side chains, and here in red, polar or charged side chains. And that mix enables an aqueous solute or substrate to pass through the channel without getting stopped from moving through that membrane, right? So it's a favorable environment for an aqueous molecule that's used to being in an aqueous solution to pass through it. So like a salt, any ions would need a channel, usually made up of multiple alpha helices that forms a pore. But also water, aquaporins, form multiple spanning, it's a multiple transmembrane spanning protein that forms a pore that allows water to move through. So water will naturally diffuse through slowly across the membrane, but in order to really balance the osmotic or osmolarity of the cell and maintain it, it needs to move faster, and so aquaporins help the cell to do that. Also for ions, these channels can allow them to get across what they would not normally be able to cross, that hydrophobic region of the lipid bilayer. So remember, even though ions are small, they're charged, so they can't get across by diffusion like other small molecules like oxygen or carbon dioxide. These transmembrane proteins, as I mentioned, don't have to be alpha helical in their structure. They can have many beta sheets that assemble and are oriented, again, having hydrophobic side chains in the core, that's the membrane spanning region of the protein, and the hydrophilic side chains here in the less organized portion of the protein, or on the interior of that beta sheet. So one example of this type of transmembrane protein that has many alpha helices that span the lipid bilayer is rhodopsin, and it helps the eyes to detect light. It has a region where it can bind retinol, and it pumps protons out into the extracellular space. And this gradient that it forms with the protons can help with the signaling to our nervous system. So it helps us to detect the amount of light that's coming into the eye. Also, cell shape can be maintained by membrane-spanning proteins. These are of a different type, the attachment proteins that can attach to transmembrane proteins. So this transmembrane proteins can hold on to other proteins just inside the cell, under the surface of the plasma membrane, and hold a whole network of cytoskeleton, spectrin, and actin in a certain orientation to maintain the shape of cell, for example, the biconcave shape of a red blood cell. And that shape is important for the function of being able to carry as much oxygen, as much hemoglobin as possible. Okay, so that's all I want to go over for Chapter 11. And are there any questions before I go on about the lipid structure, the types of lipids that are found, the fluidity of the membrane, that kind of maintenance of the orientation of the two layers? And you should know the types and the functions of the membrane. So Chapter 12 goes over the properties of permeability of membranes, lipid bilayers. Transport focuses on a specific type of protein found in, or a specific group of proteins found in membranes, and those are the transport proteins, which are transmembrane proteins that form channels or act as transporters. And it goes over the mechanisms of movement of solutes either in or out of by passive means or active means. And it also goes over some examples of how these channels and transporters are important to function of certain cell types and in general carrying out the movement of molecules down a gradient, against a gradient. The gradient can be electrical or chemical, and depend on not only on the charge of what's being transported, but also what's the charge of the inside of the cell versus the outside of the cell, or the concentration of solutes in versus outside of the cell. So examples are given of the movement of water, and also a lot of time is spent on the movement of ions, such as sodium, which is really key, has a key role in so many other functions in the cell to maintain not just the solute, not just the electrical membrane potential of the cell, but also to help maintain pH, salt concentration, and to help bring in other important molecules into the cell by creating a gradient that can help to move sugars like glucose into the cell. And then at the end of the chapter, it goes over the specific example of the function of not the sodium transporter, but the sodium channels in the function of nerve cells in how they signal along the axon and also at the synapse. So, we all know membranes are selectively permeable. What does that really mean? Certain things get in, certain things don't get in, or certain molecules get out while others don't freely get out. But just breaking it down, small molecules can easily pass through without any help of a protein or energy. Okay, so they can all get in or out. And so what is their movement going to be determined by? What? Concentration, a gradient, right? So they're going to naturally move from an area of higher to lower to get that equilibrium. Okay? So small molecules, gases like oxygen, carbon dioxide, nitrogen, small molecules like benzene, even water, even though it's polar, can get through across the membrane lipid bilayer, and small molecules like ethanol and glycerol, even though they are polar, they're not charged, and so they can, slowly get across the membrane. If they're larger polar molecules, even if they're uncharged, they cannot. They're too big, they need help. even as I mentioned in the case of water, in order to quickly move water back and forth, then there is a protein that helps with facilitated diffusion in that case. But the larger molecules like amino acids, some of which we know are polar, some of which we know are charged, large molecules like monosaccharides like glucose, and nucleotides, which the cell obviously needs to get in order to carry out its metabolism and really for its survival, these need help of a protein to get into the cell. And same thing with ions. So membrane proteins, these transmembrane proteins, are what helps these larger or charged small molecules to get in or out of the cell. And that applies not just to the cell membrane, but also to all of the compartments. There are certain compartments that want to build up a concentration of ions, for example, in the case of the lysosome, that has a more acidic environment. In order to maintain that more acidic environment, it has a transport protein that helps to move protons into that compartment. So there's a lower pH here in the lysosome than there is in the rest of the cell, which has to be maintained at neutral pH because what needs to function inside the cells at neutral pH? Why would the cell need to be at neutral pH? Enzymes, yep. Same thing with the sodium concentration, has to be much lower inside the cell for enzymes to work than it is outside the cell. And in the case of energy production, there's a certain direction that, for example, glucose or pyruvate has to be transported in order to get that ATP made. And there's also a proton gradient that's established, so an imbalance of protons across that inner mitochondrial membrane in order to facilitate the oxidative phosphorylation of ATP. So there are really two types of transport that occur with the help of proteins. There's passive transport, which doesn't require energy, and active transport, which does require energy. And in both cases, they're transmembrane proteins that are aiding in these processes. But the main difference is that active transport will require some form of energy, either ATP or light energy, in order to occur. And with passive movement, you always have to take into consideration the concentration gradient that exists on both sides of the bilayer. So there are two classes, what your book used to call carrier proteins, but now, and I noticed it has changed to calling them transporter proteins or transporters, and then channels. They're both transmembrane proteins, but they work in a little bit different way. The transporters are very specific for a single or very few solutes. So they have a specific shape that determines what they're able to carry across the membrane, whereas the channels are size exclusion proteins. So they determine based on size and also charge what they're going to let into or out of the cell. So both of these proteins work in a direction, allowing directional movement. In the case of the transport protein, because of that specific shape because it's based or able to carry a substrate or a molecule based on its shape or its fit in that protein. These are actually allosteric, so that shape will change. Okay, once the solute has been bound, it will change and allow a different shape to kind of open the gate. So on one side or the outside of the lipid bilayer, a protein, transport protein may naturally exist unbound, open to the outside. Once the solute binds, it closes that gate and opens the gate on the inside, allowing the solute to move into the cell. If it's moving down a concentration gradient, this will happen naturally without the input of energy in a passive manner. Okay? The sodium pump is a transport protein. It's actually sodium-potassium pump is an example of a transport protein. A really key transport protein in the cell because it's maintaining that not only the salt concentration being low inside the cell, but also the membrane potential. So all biological membranes have a net negative charge on their interior and a positive charge on the exterior. It also allows for the coupling of transport of sodium, which requires ATP, because the gradient, the movement of sodium is against the gradient, so it's an uphill reaction we're talking about, unfavorable. And with the movement of potassium, which also is moving against its gradient. And this pump works in a stepwise manner where if the first step doesn't happen, then the subsequent steps won't happen. So if that first sodium binding step doesn't happen, then there will be no transport of potassium. Yep. So out of all the ion concentrations, there's a net positive charge outside of the cell. Net negative charge, I think it's negative 70 millivolts, is the resting potential of the membrane of a cell. So if you actually take out a chunk of membrane, put in a little micropipette, and measure the voltage across that membrane, which a lot of physiologists do to understand that what's going on in various physiologic reactions inside cells, you will see without any action of the pump, there's or any channels being open, there's just a net positive charge on the outside, net negative charge on the inside. So it doesn't really show it here, but there are actually 3 binding sites on the pump where sodium can bind, and that will, again, with the help of the input of energy, the ATP actually phosphorylates the protein pump, and a conformational change occurs to open and release the sodium to the outside of the cell, and at that point, the potassium will bind to its site, again, a specific site because it's a transport protein. It's not just based on ion size or charge. It's actually a specific interaction with the shape of the ion, with the shape of the protein. And then that that will trigger, once both of the sites for potassium binding are filled, another conformational change will result, and the protein will become dephosphorylated, and the potassium will be released to the inside of the cell. So here are the steps in the activity of the pump. So first, 3 sodiums bind. ATP phosphorylates the protein, which triggers a conformational change. The 3 sodiums are released. 2 potassiums bind. Once those 2 sites are occupied, another conformational change occurs when phosphate group or dephosphorylation occurs of the transport protein. And once it's dephosphorylated, then the potassium is released after that conformational change that occurs as a result of the loss of the phosphate group. So there are other enzymes that are helping to catalyze this dephosphorylation. It's based on the shape of the ions, right? So it's a specific interaction of the sodium with so it's in these images, they just show it as a different shape of the ions. It's a different size, shape, yes. And it's interacting with different sites with protein too. You could see as just kind of the sides of the protein. So they don't bind to the same area of the protein. Okay, so why are sodium and potassium pumps so important? As I mentioned, they help to maintain the amount of sodium inside the cell at low levels. And they help also because they help to, when coupled with a proton transmitter, proton transporter protein, help to maintain the pH or the balance of protons inside the cell. They also, when coupled with a glucose transporter protein, help to bring nutrients like glucose, amino acids, other large molecules into the cell. In the nervous system, they're important for all of these functions. But another type of sodium channel is important for the action potential. So it's not the same as the sodium-potassium pump. But let's just look at how the sodium-potassium pump maintains some of these important features of the cell. So water, as I mentioned, it can diffuse into and out of the cell slowly. If it naturally leaks in, over time, the pressure within the cell, the water pressure, would build up and cause the cell to burst. Why is that? Why is water naturally moving into the cell? Well, osmosis, but what determines the direction of the water movement? Concentration, right? So the cell wants to have a lot of molecules in it so it can carry out all these chemical reactions, whereas outside the cell, it's unlikely that you'll have such a great high concentration of solute. So the water naturally wants to move in to make equilibrium as far as the solute concentration. So osmosis is just a word we use to describe that natural movement of water to balance the solute concentration. If the solute concentration is really high outside of the cell, then the water will move outside of the cell to balance and get it more concentrated inside the cell to balance that. So you saw that in one of our first labs when we were looking at the plant cell in salt. So in order to help to balance that, sodium ions are pumped out in order to help balance that solute concentration inside and outside of the cell, and also to balance them and prevent and cause the movement of water. Aquaporins also help with the movement of water outside of the cell. Another reason that the sodium-potassium pump is important is that it creates a gradient of sodium that can be used by other transport proteins to bring in solutes. So while the pump itself is not transporting glucose, it can through a symport. So there's different ways that solutes can get into the cell. They can get in through protein that carries out just the movement of one uniport. Just one molecule at a time. Or it can carry two molecules at a time. So down, so remember that sodium is higher inside than outside of, higher outside than inside of the cell, so it would naturally move down its gradient from an area of high concentration to low concentration. So that's a favorable movement of the sodium. But along with that, it can bring in glucose, which would bind specifically to the transport protein in this case, bringing both in to create an energetically favorable movement of glucose into the cell. And then you can also have an antiport where things are moving in different directions. One down a gradient, even if the other is moving against its gradient. Just see if I forgot to bring it today. Some of these animations are worth watching, but I forgot to bring my CD today. So the sodium-potassium pump also can help with movement or signals that are essentially nerve signals. So in the Venus flytrap, once a fly triggers consecutively three of the hairs that are on the inside of that part of the plant, and the sodium-potassium pump will cause an influx of potassium such that it uses that gradient of sodium moving down its gradient to signal for the plant to close. So the sodium-potassium pump in this plant creates a gradient of a lot of sodium outside of these cells, low sodium inside the cell. When there's a trigger that opens a channel that allows the sodium to go down its gradient, then it signals for the closure of the leaves of the plant and the trapping of the flies. So in a similar way, our own neurons can use the gradient that's created by the pump to signal along very long distances, okay, even up to a meter long, from one part of our body to another through the axon of the nerve cell. So what does that really mean? That means, I'll just briefly go over it in the last few minutes that we have, and then we can go over it in more detail later. So that means that this gradient, this membrane potential, this resting membrane potential of having a net negative charge inside the lumen of the axon, inside the cell, in the cytosol here along the axon, versus the positive charge outside of the cell can be used by another transmembrane protein, an ion channel, like the sodium ion channel. Now this is a different protein than the sodium-potassium pump, which is a transport protein. This is a channel that is moving ions based on size and charge, and it's specific for sodium. When there's a resting conditions, the cell has a net negative charge inside. Sodium wants to diffuse out. Diffuse in because there's a higher amount of sodium, a large amount of sodium outside of the cell. Okay, so out versus in. So it naturally wants to move to an area of lower concentration. And it can't diffuse through the membrane, but it can go through a specific channel, the sodium channel, when that gate is open. So when it's open, it allows for the specific substrate, in this case the sodium, to pass through. Closed, nothing can move through, or if it's inactivated in some way. But once it opens, you get an influx of the sodium changing the charge. So previously it was negative, changing the charge at this point, and flipping the charge just in that region of the transport, of the channel. Okay? So along the channel you start to get the reversal of the membrane potential, which we call an action potential, where you get the inside being more positive rather than more negative. And it's through the, see it's here, but once the channel is open, here by the yellow arrows, you get that influx of the sodium ions, causing that region of the axon to become more positively charged internally. Now, as soon as those channels then close again, the resting potential will return. So you'll get the maintenance by that sodium-potassium pump, which is different than the sodium channel, will return the axon at that point to the resting potential, or that net negative charge. Let me see how much time. So what happens is there's along the axon there are hundreds of these channels that will allow for the depolarization at a local site and allow for that change in the polarization from the resting to the depolarized state to move along the axon. So the propagation of the signal is only in one direction due to these inactivated state of the channel, unable to then open and let more sodium through. Okay, so it's closed, it becomes open, it becomes inactivated. At that point, the other types of proteins, the sodium-potassium pump, will reorient the gradient of sodium and potassium so that you have the resting, the original resting potential. So very quickly, because the influx of the ions can happen in a matter of milliseconds, very quickly this depolarization can occur, and very quickly it can occur along the entire axon. And this is really what the nervous system is, what is regulating our ability to move, to think, to learn, to remember, all of these functions that make us what we are as humans, human organisms. So this cellular event of these channels and the pumps together working to change the polarization across these membranes is really what's key for these functions that we take for granted. So we talked about nerve signal. Nerve signal, actually, there's a lot of channels and transporters that are involved in neurons and nerve cells. But one of them that allows the nerve to signal over long distances is the sodium channel that we just talked about, coupled with the activity of the potassium channel, a different channel protein. Naturally, these sodium channels and other ion channels like the sodium channels flickering between open and closed. But most of the time at the resting potential, they're closed. So we're going to start in that position. When there's a stimulus that causes a change in the balance of those charges, there are two things that can happen. The first initial change we call a depolarization. So from our minus 60, minus 70 millivolts of the resting potential, an influx of some sodium ions causing the charge to, or the resting potential to rise, at which point it will either stop, or if it reaches the threshold, which I think is around minus 40 millivolts, if it reaches that threshold, it will continue to stimulate the opening of other sodium channels. Dramatic change in the voltage at that site along the membrane of the axon. So this is not the entire cell suddenly flipping, it's only at that tiny site along the axon where these channels have opened. At the peak, then, the sodium channels lock as they're open for a period of time, and then they lock. And then here's where the potassium channels come into play. As they are triggered to open by this action potential, they let the potassium then out of, because there's a large amount of potassium on the inside of the cell, thanks to our sodium-potassium ATPase. Okay, so again, dropping the voltage across the membrane until it reaches the resting potential.
Yeah.
It does. It only travels in one direction. Why doesn't it travel back in two directions? The lock, right? So it's locked. It takes time for the sodium channel to recover, and by that time the signal is way down to another part of the membrane, so it's not going to be triggering the signal in the other direction. That locked is an important step in the function of these channels. Then at the terminus is going to be signaling another neuron how to interpret the feeling or that signal from the sensory neuron. We start with a sensory neuron, we end with a motor neuron, picking our arm off the hot stove or off the sharp pin or whatever. So what happens at the end of the nerve terminus? Because there's another set of channels that are involved, but they're not they're not triggered by the changes in the voltage. It's the synapse, so that small space between the terminus, or the ends of the axon, and the next body of the next neuron, which releases, it will, the action potential will trigger vesicles carrying neurotransmitters to release their contents into that cleft, and those through a ligand specific or ligand dependent channel will then propagate the signal, which again depolarizes the membrane and can start another action potential in the next neuron. Okay. So is everyone clear on the difference between the voltage-gated sodium channel and the sodium-potassium ATPase? Even though they're located in the same membrane, they have very different functions. So this is kind of always on, always busy, and the sodium channel not always on. Has to have that voltage change to be triggered, whether it's on, open or closed.